Allah hafazakum. Okay, I'll talk about the immune system in gastrointestinal tract, but there are similarities between the mucosal surface and skin. If we have time, I'll talk about immune response in the skin as well. There is a very important concept about the epithelial barriers. It's the same for all barriers. It's not only for intestinal barrier. Please have a look here. Here. Oh my God. Oh. Epithelial cells in all barriers. Epithelial cells in all barriers express innate immune receptors. دکتر علی این برام یه سر بزن با هم. این آخرها خستن قلقلکشون هم بده. قلقلکشون بده چیه بگو. میدونم ولی نمیتونم چجوری تازه کنم. Okay. All epithelial cells.
Das Namen mit der Klasse, Intensive, Surgeista.
Epithelial cells of the barriers. All of them. Please have a look here. They will express innate immune receptors. All epithelial cells express epithelial innate immune receptors. What does that mean? Epithelial cells of the barriers express immune receptors. What does that mean?
Not self.
To detect non-self. Yes, it's okay. Epithelial cells can detect non-self because they have receptor. But what does that mean?
Respond to pathogens directly.
Yes, epithelial cells, we have different types of epithelial cells. You know from histology and other courses you have or you might have. Epithelial cells indeed are similar to innate immune cells. As you remember, the first line of immune response is mediated by innate immunity. Do you remember? Innate immunity is the first line of immune response. Clear? Do you have problem with this one? With this sentence? Innate immunity is the first line of immune response. And epithelial cells are the first cells that are exposing to the antigen. They are barriers of the body. If any pathogen, any non-pathogen, any microorganism wants to go inside the body, they should pass the epithelial barrier. So the epithelial cells lining the barriers are the first cells that can detect the microorganisms with their receptors. So it's because they have the receptors, PRR, pattern recognition receptors of innate immunity. So epithelial cells can detect PAMPs, pathogen-associated molecular pattern. So, what do you know of inflammation? Do you remember anything about inflammation?
A lot of the mechanism.
One aim of inflammation. What's the aim of inflammation? One sentence.
It's immune response.
Immune response. What else? More exactly.
Innate immune response to infection in the cell.
One function of innate immunity. Okay, what else? What's the aim of innate immunity? What happens in inflammation? Sorry, what's the aim of inflammation? What happens in inflammation? Whenever we have inflammatory response, what is going on and what's the aim?
Any damage occur, there is a response of the immunity. For example, exit the macrophage and enterocyte from the circulatory system to the tissue, and after that,
Calling of the cells to the tissue, infected tissue, recruitment of the cells of the immune system to the infection site.
Try to prevent the bacteria or virus from separating into the body.
Again, please.
Prevent the bacteria or virus from separating into the
No,
body.
inflammation means what your classmates said. Inflammation means recruitment or calling of the cells to the site of infection. For example, if I have an infection here, the first event of the infection is induction of inflammation. The first event of all immune response is induction of inflammation. And it is started by pro-inflammatory cytokine. Do you remember pro-inflammatory cytokine? TNF alpha. Have you heard it? TNF alpha, interleukin 1 and interleukin 6. They are 6, sorry. Interleukin 1 and 6 and TNF alpha are 3 pro-inflammatory cytokines. They are pro-inflammatory cytokines. Indeed, they are inducer of inflammation. What does that mean? For example, if I have an infection here, a start of infection here, the epithelial cells are the first cells that can detect the infection. Am I clear? The epithelial cells are the first cells that can detect, can sense the infection. And the epithelial cells and other cells located in this place, for example, macrophages and dendritic cells, mast cells, mainly macrophages and epithelial cells, are the first cells that detect this, the infection. The first thing that these cells should do is the calling of other cells. The epithelial cells and macrophages cannot induce by themselves a strong immune response. They should call the other cells to come to the site of infection. They should recruit the other cells to the site of infection. So the epithelial cells and macrophages will produce cytokines, pro-inflammatory cytokines. That pro-inflammatory cytokines activates a process which is called inflammation. And as a result of this process, inflammation, the other cells will come to the site of infection. So without inflammation, we do not have any calling. We do not have any informing to the other cells. For example, if there is an infection here, bone marrow, how bone marrow should be informed about the infection. The bone marrow, how should be informed about the infection. In the site of infection, we will have pro-inflammatory cytokine. These cytokines go to the bone marrow, and inside the bone marrow, tell to the bone marrow that we need, for example, neutrophil, we need lymphocyte, we need monocyte. They call other cells from bone marrow to the site of infection. So, epithelial cells are the first cells of the body that can detect the infection, and then they will produce, after binding to the antigen by the receptors, PRRs, they will produce pro-inflammatory cytokines in order to activate inflammation, and they call other cells to the site of infection. So this is one of the functional barriers, calling of other cells or activating of inflammation. We have some specialized epithelial cells like goblet cells that produce mucus. And we have another specialized epithelial cells like Paneth cells that produces antimicrobial peptide. When we say there are several antimicrobial peptides that they bind to the microorganism and kill them. Antimicrobial peptide. They are called natural antibiotics. They bind to the surface of the bacteria and they can lyse the bacteria or other microorganisms. We have different kind of antimicrobial peptide for different kind of microorganisms. they are produced mainly by Paneth cells, and some other cells can produce them, like neutrophil and macrophage. And some specialized epithelial cells samples the antigen of the barriers. I'll talk about it in next slides. You can see here the structure of a small part of the intestine as a sample of gastrointestinal tract. You can see here the epithelial barrier, one layer of epithelial cells. Here is the lumen of intestine. It means here is the inside of the intestine, and then we have epithelial cells. We have connective tissue, the first layer, epithelial cells, second layer, immune cells. The second layer, scattered immune cells in the connective tissue under the epithelial layer, and draining lymph node as a third layer of immune response. I said we have 3 layers of immune response in the barriers. First layer by epithelial barriers, second layer by the cells that are scattered in the lamina propria, in the connective tissue, and third layer by the draining lymph nodes, cells that are located, mainly B cells and T cells, inside the lymph nodes. So you can see here that, as I said, we have Paneth cells that they produce antimicrobial peptide. You can see here the Paneth cells that produce antimicrobial peptide. They are specialized epithelial cells. You can see goblet cells that they produce mucus, and we have a lining of mucus on the epithelial cells. Besides, we have M cells that their role is antigen sampling. As I said in previous slide, antigen sampling. M cells are located above a structure underneath the epithelial cells. That this structure is called Pierce patch. They are similar to a lymph node. Not, they are similar. They only have follicles. They are similar to cortex of the lymph node. As you remember, in the cortex of lymph node, we have follicles. That inside the follicle we have mainly which cell? Inside the follicle, which cell we have mainly? Which of the cells of the adaptive immunity? B cells. B cells. Coronal expansion, germinal centers, primary follicle, secondary follicle. Do you remember?
Yes, yes.
Follicles are a compartment inside the cortex or other site of the body that they are mainly the main site for B cells. So inside the Pierce patch we have several follicles, and inside the follicles we have B cells. And M cells will transfer the antigen from the lumen toward the Pierce patch. They will transfer the antigens toward the Pierce patch. And the cells inside the Pierce patch, like the dendritic cell and B cells and other cells, can recognize, can capture the antigen. If the antigen or is from pathogenic bacteria, they will induce an inflammatory response. They will induce the proliferation and production of antibody. But if there is no pathogenic microorganism, we won't have any inflammatory response. So we have Pierce patch as a lymphoid tissue beneath the epithelial cells, and we have M cells above the Pierce patch, that transfer the antigen inside the Pierce patch and follicles. Besides, we have some intraepithelial lymphocytes. Intraepithelial lymphocytes. That they are mainly CD8 positive. That, as you know, CD8 positive T cells are cytotoxic T cells. We have between the epithelial cells intraepithelial lymphocytes that intraepithelial lymphocytes there is a phone over there which is ringing. Nothing more. So, between the epithelial cells, we have intraepithelial lymphocytes that they are mainly CD8 positive. As you remember, CD8 positives are cytotoxic T lymphocytes. So, if any virus or bacteria can infect the epithelial cells here, these T cells can recognize them and destroy them. Indeed, CD8 positive intraepithelial lymphocytes try to eliminate source of the infection. Let me explain it again. A bacteria tries to come inside the body. They first infect the epithelial cells or virus and try to proliferate, multiplicate inside the epithelial cells and then invade other cells. But the T cells, intraepithelial T cells, will kill the infected cells in order to eliminate the source of the infection. Besides, they will produce some kind of cytokines to inform other cells of the body, to inform other cells of the tissue that there is something harmful, for example, here. What's the main role of cytokine?
Transport the signal between the
Signal transportation or communication. Cells of the immune system needs to talk to each other. And one mechanism of communication between cells of immune system is production of cytokine. So intraepithelial lymphocytes and epithelial cells will produce cytokines in order to inform other cells that something abnormal is going on here. It's an information. So, for epithelial layer, it's enough production of mucus, production of antimicrobial peptide, induction of a physical barrier. We have tight junction between the epithelial cells that prevent invasion or entrance of the microorganisms, and having intraepithelial lymphocytes as well. They are the main function of epithelial barrier. But the second layer is the cells that are scattered throughout the lamina propria or connective tissue under the epithelial cells. We have different kind of cells here. As you can see here, most of the plasma cells of the mucosal system, most of them will produce IgA. As I said, IgA is indeed the antibody of mucosal surface. And it is dimer, which is produced by plasma cells in the connective tissue, and the IgA will transfer inside the lumen, inside the intestine, in order to neutralize the bacteria inside the intestine. For example, if there is a bacteria try to come toward the epithelial cells, this antibody will bind to the bacteria and prevent the binding of bacteria to the epithelial cell. So it's a neutralizing effect of IgA. As you remember, I said IgA can neutralize bacteria, virus, and their toxins in the mucosal surface. How IgA will bind to the microbes and prevent its binding to the epithelial cells. And besides, we have T cells in the connective tissue. The main T cells of connective tissue are T helper 17 and T regulatory. T helper 17 and regulatory T cell. What do you know of these cells, T helper 17 and regulatory T cells? What do you remember?
Regulatory about stop immune response.
The function of regulatory T cells is
Stop immune response.
suppressing or stopping the immune response. How a Treg can suppress immune response?
To protect us from autoimmune.
To prevent autoimmunity or unwanted immune response against microorganisms. It's the aim.
Release signals.
It's the aim of regulatory T cell. But how immune regulatory T cells or Treg regulatory T cells can suppress immune system?
By-
You said the aim of the regulatory cell.
by activate a new innate immune system.
By production of cytokines TGF beta, yes, and IL-10 to suppressor cytokines of the body. Regulatory T cell will produce normally whenever there is no need to inflammation. Whenever we have only commensal or normal flora here, there is no need for immune response. There is no need for inflammation. So T regulatory will produce TGF beta and IL-10 in order to suppress other cells. But whenever we have a pathogenic bacteria, the story will change and we will have inflammation. Besides, TL17 will produce a very important cytokine, IL-22, interleukin 22, IL-22, that main function of interleukin 22 is induction of helping to mucosal surface immune system by induction of antimicrobial peptide production by planet cells, IL-22, when bind to the epithelial cells, induce the production of or increase the production of antimicrobial peptide. Besides, it increases the tight junction between epithelial cells in order to prevent the invasion of bacteria. So the main T cells in normal connective tissue of the intestine are TLR17 and regulatory T cells. Besides, we have different kind of dendritic cells, DC, dendritic cells in the connective tissue. That some of them in normal situation, dendritic cells of the connective tissue are tolerogenic. Indeed, they induce tolerance. They are different from normal dendritic cells. So in normal, I think I have a slide. Okay. Until now, I talked about the immune components of the first layer and second layer in summary. And if the first layer can prevent the infection, we won't have any response in the second layer. I talked about this repeatedly. We have 3 layers of immune response: epithelial layer, connective tissue, scattered immune cells, and draining lymph nodes. And each step going forward to each one step going forward, we will have a stronger immune response. But if the first layer, epithelial layer, can prevent infection, we won't have any immune response in connective tissue. But if connective tissue can prevent, we won't have any immune response in lymph node. But if both of them cannot prevent the infection, we will have induction of immune response inside the draining lymph node, nearby lymph nodes, that we have T cells and B cells here, adaptive immune cells that will induce a stronger and more specific immune response. Question, comment?
No, doctor.
You can see, I have forgotten the function of one important cell. Oh my God. We have some specialized dendritic cell in the intestine that they do sampling of antigen. We have 2 ways at least of antigen sampling of the intestine. One of them is mediated by antigen sampling. The one way that I talked about was M-cell, that they sample the antigen of the intestine. Another way is mediated by dendritic cells. Dendritic cells will capture the antigens of the intestine and will transfer them to the nearby cells. You can see You can see here, this is the epithelial cells. These are the epithelial cells that dendritic cells can capture the antigen. Here is the lumen of the intestine. They capture the antigens of the lumen and they will present the antigens to the nearby T cells to activate them. This is antigen sampling of the intestine. Another way of antigen sampling was made by M cells. This is an M cell that can transfer the antigens of the lumen inside the Pierce patch. And we have different cells besides the M cell. Another function is production of IgA, as I said, plasma cells that produce dimer IgA. IgA will be transferred to the lumen, and inside the lumen, which is called secretory IgA, this secreted IgA will neutralize the bacteria and their toxins. And besides, we have a layer of mucus on the epithelial cells, that inside the mucus we have both antimicrobial peptide and SGA. So you can see here that they are the microorganisms of the lumen, they are the epithelial cells. We have a free space between microorganism and epithelial cells. You can see here. This is the epithelial layer, and this is the microorganism of the intestine, but there is an empty space between the microorganism and epithelial cell. This empty space is mucus layer. And we have antimicrobial peptide, AMP, antimicrobial peptide, and secretory IgA inside the mucus. If any microorganisms try to come toward epithelial cells, the antimicrobial peptides try to kill them. Besides, secretory IgA try to bind to them and neutralize them, prevent they bind to the epithelial cells. So we have a mucus layer containing antimicrobial peptide and SGA. Question, comment? Okay, we have two types of immune state. We have two types of immune state in the barriers of the body. One of them is called home.
Move it. It's have any issue.
We have two states of the immune response inside on the barriers. One of them is homeostasis, normal immune response, and another one is inflammation. Whenever there is no pathogen, but there is macroflora, there is normal flora, commensal bacteria, we have mainly tolerogenic dendritic cells and regulatory T cells that they produce IL-10 and TGF-beta. So in homeostatic response, we have only sampling and assessing. Immune system constantly is sampling and assessing the content of the lumen inside the intestine, And if there is no pathogenic microorganism, we will have tolerogenic TC activation, production of IL-10, or T regulatory function, production of TGF-beta and IL-10, that these cytokines suppress the activity of other cells. Whenever we have pathogenic bacteria, we have inflammation. And when we have inflammation, there is no suppressory cytokine host cells. We have activated dendritic cells, we have T helper 1, T helper 17, interferon gamma, inflammatory cytokines, TNF alpha, and activation of other cells of the body. So there are two different states of immune. in normal situation or homeostasis, sampling and assessing, but if there is epithelial damage or epithelial damage is a sign of invasion. In normal situation, we do not have epithelial damage. So whenever we have epithelial damage, it means that there is an abnormal event over there. So in epithelial damage or any abnormal situation that is mainly induced by pathogenic microorganism, we will have activation of inflammatory cells, inflammatory cytokines. They are called anti-inflammatory state. Here we have inflammatory state. So in most of the time, we have anti-inflammatory response. Question? If we have unwanted immune response against normal flora microorganisms, some diseases may occur. For example, have you heard anything about IBD, inflammatory bowel disease? There are two kinds of IBD, Crohn's disease and ulcerative colitis. In both of them, Crohn's disease and ulcerative colitis, we have unwanted immune response against normal flora. We have unwanted inflammation inside the gastrointestinal tract. They are different, but the mechanisms are same. But the main reason for the beginning of them is unknown. We do not know immune response against which microorganisms occur, but there is unwanted immune response against normal flora in both of them. And whenever we have unwanted immune response against gluten, antigen of the food, we would have a disease which is called celiac disease. It's similar to both of them are similar to autoimmune disease. Unwanted immune response against normal flora and food antigens. And sometimes we will have allergic reactions. Allergic reaction against food antigens. That it is again unwanted immune response. Indeed, if we do not have controlling of it, it's better to say, if in the barrier immune system is not controlled, is not in the homeostatic state, and we will have unwanted immune response against microorganisms. Indeed, there is a problem in the discrimination between pathogenic and non-pathogenic. So in this patient, we do not have discrimination between pathogenic and non-pathogenic, and we have unwanted immune response against normal flora or against food antigens. Let me skip the summary. Please have a look here. Microbiota and the immune system dialogue. As I said to you, there is a dialogue between microbiota or normal flora of the barriers and immune system. And I said that without normal flora, without commensal bacteria and other microorganisms, we won't have normal immune system. For example, in germ-free mice that are grown in a sterile condition, as I said, we have reduction in secretory IgA and serum immunoglobulin. So anybody, mice that are grown in germ-free condition, they are grown in a sterile condition, they have less amount of serum immunoglobulin. So they have immune deficiency. Or sometimes they will have reduced T cell number. They will have reduced cytotoxicity of CD8 positive cells. Indeed, they cannot kill their target easily. And some other deficiency of immune system. So without normal flora, we will have deficiency in immune system. We will have defect in immune system. We do need normal flora in order to have normal immune system. The cutaneous immune system is similar to This is the one. There are so many immunodermatological disorders. So many. All of them are because we have unwanted immune response. Uncontrolled immune response on the skin. Some of them are autoimmune like diseases. There are many of them that we do not have to check all of them. But the structure of the skin, we have multi-layer of the epithelial cells, we have intraepithelial dendritic cells. You can see them, which they are called Langerhans dendritic cell. Langerhans dendritic cell will capture the antigen and take the antigen to the nearby lymph node. It's the normal route of immune response, taking the antigen by dendritic cells to the nearby lymph node and presenting the antigen to the T cells, and activation of the T cells we will have. So we will have intraepithelial dendritic cells, which they are called Langerhans cells. Besides, we have antimicrobial peptide again here, different kind of antimicrobial peptide that are produced by epithelial cells, and again we have scattered immune cells in the connective tissue. But again, like similar to intestine, in normal situation we will have TGF-beta and IL-10 by Tregs and other non-tolerogenic or non-inflammatory cells. So in normal skin, like normal intestine, we do not have immune response against microorganisms or normal flora. But again here, we have intraepithelial lymphocytes, we have dendritic cells, we have scattered in 3 layers of the immune system. You can see 3 layers: epithelial layers, connective tissue layers, and draining lymph nodes. And we have a scattered immune system throughout the connective tissue. And you can read them. That's it. I have nothing more. And they are